Yes, so the idea of this webinar is to sort of continue on the last one, and to move on to a population level analysis of the deep sequencing data that we have also painstakingly collected over the last 5 years. So this has been work that's been going on for the last, gosh, I don't know, more than 2 years. And it's taking quite some time to get up to speed, and so I'm very happy to share with you the results from scaling up the application of Phyloscanner to a large population-based sample, and specifically we focused on one question, which was high prevalence fishing communities on the shores of Lake Victoria, the predominant sources of new infections in the broader region, further inland, away from those fishing sites. So that's the topic, that's the question. As you can imagine, there were a whole lot of people involved in this, and I will, and I hope I'll make sure to acknowledge the right people at the right time during my presentation. So for those of you who don't know me, this is a photo, given that I switched off the video. But you can see, like, I'm not sure if you can see the mouse. So this is our oldest, Jonas. He's now almost 3. And yes, so I have a little family. I did my PhD in 2009. This was in statistics and actually network science. So I'm sort of getting back to my origins in a way. And I've done HIV phylogenetics since 2013. Initially on Dutch data. But then, so when PANGEA came around, this was very obvious that this is the next choice and a train to get on, and it'll be so much more relevant and exciting to do this work on data from Pakai, but also other places in Sub-Saharan. And then since 2017, I have my own group here at Imperial. There are 2 PhD students and 1 postdoc. Okay, just to set the scene, I hope that you're all still with me. So if there's any issues, please just kind of wave or let Lucy know. Otherwise, I will just continue like this train that never stops. So to set the scene, as you all know, and this is the figure from Sarah Jane's paper in The Lancet, that the HIV epidemic is markedly heterogeneous, and there are these small areas of high HIV incidence that we all know as hotspots. And critical to current prevention programming is the idea that geographic targeting of combination HIV prevention to these hotspots is essential, and that's for 2 reasons. First, to, of course, make a cost-efficient use of resources, but also to substantially ameliorate the broader epidemic. So indirect effects that the direct intervention in these relatively small areas has. In East Africa, one example of these hotspots are fishing sites. This is, I think there's a number of you on the call have actually been there. I haven't yet. So I'm talking about a place that I've actually not visited myself, but hopefully this is going to come soon. This is a photo that I was given from the lake. And yes, and since 2014, combination HIV prevention is recommended to these fishing sites by the Ugandan National ART HIV and Prevention Plan. And this recommendation was based on 2 points. First, of course, because of high HIV incidence, which I'm trying to show in these maps here. So here, amongst women, an HIV prevalence of about 40%, much higher than further inland. Hopefully you can see my mouse. And then amongst men, of course, HIV prevalence is lower, but still much higher in fishing sites compared to inland communities. So this recommendation was based on high HIV incidence in these fishing communities, but also the assumption that these fishing sites are a major source of new infection among inland communities, so those that are further inland away from the source of the lake. And the idea was that we wanted to support geographic targeting and support this intervention by confirming the assumption that fishing communities are indeed a major source of new infections among inland communities. So this was our go-in hypothesis. And when we planned the study very early on, we realized, and this is really a big group, this is not a we, this is a lot of people, and in which I only played a minor part at this point in time. So what was realized, is probably the better way to say it, was that there are two ways in which fishing sites can seed new infections further inland. So first, of course, this is person-to-person direct transmission. So from individuals who are residing fishing sites and then perhaps via short-term travel infect someone inland. So direct transmission. And then the second mechanism is that individuals migrate from fishing sites to inland communities and then start a new HIV transmission chain in these inland communities after they have migrated. So to reflect this complexity, we used a combination of data sources to address our study objective. And as you can imagine, I'm talking about phylogenetics, about migration, including these fishing sites in the cohort. So there were a lot of people involved in the Rakai Health Sciences program, Johns Hopkins, Kate Kowalski, was just fantastic and highly instrumental in but also from Oxford, Matthew and Chris and Tanya, they helped a tremendous amount. And of course, Christoph, who's leading the PANGEA HIV2 consortium. Most of the calculations were done at Imperial on the High Performance Computing Center here. And not the least, of course, the program staff and the study participants. So they were all my thanks go out to them at this point for their contributions, without whom I, as an analyst, wouldn't have been able to do anything. But just to move on and to tell you in more detail what we actually did for this population level analysis. So first, the surveillance was extended to include the 4 largest fishing sites in the area. And between August 2010 and January 2015, in total, there were 29,115 eligible individuals in inland communities, there were 36, and 8,529 in fishing sites. And so at the time of the survey, so in this period from 2010 to early 2015, 20,089 unique individuals were found in inland communities and 6,152 in fishing sites. And of those, about 99% participated in inland communities and also in the new fishing sites by whom the cohort was extended. And in the inland communities, amongst those who participated, 14% were HIV infected and 40% in fishing sites. Among those who were infected at the time, so that we're talking about 2010, about the time of first visit. At the time of the first visit, 67% self-reported to be ART naive in inland communities and 84% in fishing sites. So these are sort of some baseline numbers on how the cohort was extended to include the fishing sites. And the second part of the study was then to use phylogenetic analysis to reconstruct likely HIV transmission networks. And so I'm trying to show this here at the bottom with sequenced individuals and 2 arrows, where the arrows indicate the relative strength of transmission in one direction that we could infer through phylogenetic analysis using the deep sequencing. I'll talk more about this in a minute. But the broader picture is that of those who are reported to be ART naive, in inland communities we could sequence 63%, and in fishing sites 74%. So just to give you a sense that we're not looking at a small study that sequenced 5% of individuals or 10% of individuals. So in that context, we wouldn't be able to do what we could do at this high coverage. So just to bear this in mind, please, as we go along. But before I go into more details and to complete the cycle of the whole analysis, so the third part was that the households and the migration events were geocoded. So now I'm trying to show this here at the right part in the slides. So we knew the households and whether they are in inland communities, shown in green, in fishing sites, here in red. And we also asked about when was the latest in-migration into the cohort, so if that person had migrated recently. And so we were able to trace whether individuals had come from outside or whether they moved between fishing sites and inland communities. And with the colors, so you can see that this allowed us to interpret phylogenetic transmission networks at a very small resolution of below 50 kilometers because we had this extra data from the mapping. And we could account for the population movement. So to make this hopefully concrete, if you look at the blue individual in this transmission network here that I'm trying to show with my mouse, so this person was found in the cohort, of course, because otherwise we wouldn't have sampled that person. So he was found in one of the fishing sites, but we also knew that that person, based on self-reported data, that person had migrated in the past 2 years before his recipient, the female, was found positive. And based on that information, and we also knew that that person was found HIV positive at their first visit in the cohort. So based on all this information, we interpret the direction from the blue person to the red person as an introduction into the cohort, because we had this additional information on the migration history. So this is how we accounted for population movement in our study. One of the first results of our analysis is that population-based analysis of NGS data is feasible. So in total, we were able to sequence 3,758 individuals. So these were sent for sequencing. And of those, 2,652 were of sufficient quality for further analysis. So we published on this before, but just to give you an overview here on the right-hand side, you can see the proportion of sequenced individuals in red of the entire infected population, and you can see the size of the circles representing the total number of infected individuals found on the survey. So overall, we estimate a sequence coverage of almost 50%, and that includes individuals who were not present at the time of the survey in the denominator. Now, I wanted to talk a bit more about deep sequence phylogenetic analysis because it enabled us to infer the direction of transmission. So this is something that chronically standard techniques can't do, and this is what makes this analysis so exciting, at least to someone who's doing these, who's focusing on methodology like I am. And so I'd like to kind of give you an idea on why that is the case. And this is essentially the same as you heard last week, but at the risk of boring you, I'll attempt this once more. And also at the risk of, I know that Chris is a very good. So we're deep sequencing individuals shown here, like 4. And what this means is that we receive relatively short sequence fragments. This is like you shoot at the virus and you get these shotgun fragments of virus, and you can map those against a reference sequence. So you have those small reads mapped. And then what we do is we don't concatenate them. So that's turned out to be a bad idea. So instead, we take a window shown in pink, and then we collect all the reads that are overlapping that window. So this would be a small region in Pole, and we collect all those reads from all those individuals. And then we align those reads, and we build a phylogeny. And what you get is something shown at the bottom, where now per any one individual, we have multiple unique sequence fragments. So not as before when you do standard phylogenetics, where you have one tip per individual. Now we have multiple tips in the phylogeny per individual. And what we expected is that from the same individuals, the reads from the same person will form a cluster in the phylogeny because they are similar. That makes sense. But what we discovered was that consistently, as we move the window from GAG to ENV and NEF, so what we found is that these clusters, they have a consistent ordering all the time. Of course, there's a bit of noise, but more or less the ordering of the blue and red individual with respect to each other stayed more or less constant. And this was, I think that's the main discovery that underlies Ethan Romero Severson's PNAS paper in the simulations, and also our analysis here on real data, that this ordering across the genome is consistent. And so that gives an idea of who came first and who came second, who is ancestral and who is a child, and hence who is source and who is recipient. And when we validated this against epidata, we're consistently estimating an accuracy of around 80% with different studies. Oliver Leyendecker and his colleagues, Rebecca Rose and et al., looked at HPTN 052 pairs using a different sequencing protocol, a different computational program, and essentially they found the same false discovery rates of 1 minus 83%. So it's we're very encouraged by that, and based on these consistent findings by different groups by now, we were just ready to press ahead. I wanted to say one more thing. I mentioned that we are moving. So we're kind of. I said like we were looking at one genomic window, and this was at the pole gene. So this is the black bar here at the in the middle figure here, and I hope you can see that. So just to give you a visual impression, what we then do is we build these phyloscans. So this is where the name phyloscan, I think, comes from, because we're moving these genomic windows and we're repeating inference for each of these genomic windows, and we're making these assignments again and again and again. And so this is shown here in these kind of phyloscan plots in the middle between C and D, where you see dots that correspond to the phylogenetic distance between two individuals across the genome. So across the genome is on the x-axis, the distance is on the y-axis, and we have 3 pairs of individuals. And the color corresponds to the ordering of those two individuals in the deep sequence phylogeny. And blue means that the male came first and the female was a child. And pink means the female was ancestral and the male was a child. And gray and violet mean that we couldn't. So either we couldn't tell about ancestry or they were actually disconnected. So this is the phyloscan, and you can see how autocorrelated that is, both in terms of distance and in terms of the color. And that's the basis for our inferences. And we summarize that in terms of these transmission networks, where we indicate, where we draw edges, links between individuals that are not gray or violet, so that are not unrelated. So we only draw edges when we see a link in the data, and the direction indicates the proportion of deep sequence phylogenies that are in favor of one specific directional assignment. Okay, see, I actually had a zoomed version of that. Anyway, So we then
Sorry, can I ask a question?
yes, of course.
Um, so how do you deal with the, because this is very short reads, right? All the time you
Yeah.
reconstruct a phylogenetic tree based on a short read. How short is this read?
250 base pairs.
And like how phylogenetic errors is taken in consideration? Right?
So, what kind of error do we take into consideration? So that's the question.
Yeah, that as
Um,
well.
and so what we do, so we're moving the genomic window in overlap across the genome. And so what that in every window we reconstruct the sequence alignment, and we're looking at different reads, right? Because so what do we take into account? We take into account differences, biological differences across the genome, right? Because evolution may have played out differently in ENV than in GAG or POL, so that's one. What was sampled? Because it could be that some reads in ENV were more sampled than in GAG. We take into account some alignment issues. So because in every time we are kind of building a new alignment, a sequence alignment of reads, and then we're building the phylogeny. So if you compound all these sources of error, basically you get, so you basically get kind of these 300 reads, 300 phylogenies in total. And this set of phylogenies all has each of these phylogenies has a different type of error in one way or another. Call it noise. And so this is what you're looking at in these phyloscan plots across the genome on the x-axis here in the middle. And you can see essentially what I'm trying to show is that this is not all noise. So more or less the genetic distances, they're not smack on a certain point value, but more or less, they're not wildly different, bearing in mind that on the y-axis you see the log scale. And also the colors, they're not extremely variable. They're not all of the same color, so meaning that topological configurations change in any one tree, but it's not crazy. So yes, there is phylogenetic uncertainty, there is biological uncertainty, there is sampling uncertainty, there is alignment uncertainty. We're trying to account for this by using these scans across the genome. And overall, our impression is that it's not too bad, and we can actually make meaningful population-based inferences. Does that answer your question?
Yeah.
Okay, that's good. Okay, so I'd like to move on then. And so one of the probably more boring computational challenges was to do this analysis at the population level. And what we discovered very quickly was that we can't just put all the reads from all 2,700 individuals into one read alignment and then build this gigantic phylogeny. So that didn't work. Instead, we had to split the total number of individuals into batches, and then do a systematic comparison of batches with each other, so that every possible pair was at least queried once. So this increased the computational workload massively, but this is what we did, and it was successful in the sense that we got some meaningful output. There's probably like some room for improvements or for playing around with the parameters, but overall, our recommendation is to split up the total number of individuals into smaller batches and then run systematic comparisons. So in total, we had, for those of you who used Phyloscanner, we used almost 2,000 independent Phyloscanner runs on 70 individuals each, and that allowed us to evaluate, make comparisons of 4.5. No, hang on, these numbers are wrong. 3.5 million pairs in total across the genome. So in each of these, for each of these pairs, we looked at these phyloscans where there were at most 300 deep sequence phylogeny. So when we did this population-based analysis, at the end we found 446 transmission networks, and they contained 888 phylogenetic linkages. So here's I'm trying to show this here at the left-hand side, where most often we just saw pairs reflecting incomplete sequence coverage. In other cases, we got larger networks, and you can see that only some of the links shown in dark gray were highly supported statistically. Others had weaker support, and for our analysis at the moment, we did not consider those links with weaker support. So in total, 351 links had low support, below 60% in total. 80 of the remaining ones, 80 were female-female pairs and 81 male-male pairs. So, and this indicated to us that there are likely unsampled intermediate cases between any two linked individuals. So this is possible to have unsampled intermediates. Deep sequencing doesn't rule that out. Simply because we had incomplete sequencing coverage, and you know, there's also at some point the method is not perfect. However, I think the important message here is that we did not see any significantly more male-male pairs compared to female-female pairs, suggesting that at least in our data there was no evidence for MSM subepidemics in rural Rakhine district. This doesn't say that they don't exist, but we didn't see them in our data that we had available. So what remained were 376 male-female linkages that were highly supported, and in 78% of those we were able to tell the direction with confidence. So this addresses the question that, you know, what uncertainty did you take into account, and how reliably can you infer the direction of transmission? And this didn't work in all cases. In 22% inferences remained ambiguous, and we only considered the 293 cases forward in our analysis. This slide is about phyloscanner deep sequence analysis among couples. And it's one of two slides that need to be considered together. So what I'm showing here on this slide on the x-axis is the median distance between the two partners in a couple, from 0% on the left side to 50% of differences in substitutions per side on the right-hand side. And the y-axis gives us the number of partners for whom a specific average distance was seen, and the color tells us about the topological relationship in those couples. So you can clearly see that among couples, the phyloscanner analysis can clearly separate the couples into a group that was closely related and a group that was distantly related. So a very strong separation property in the two groups if we have prior information on sexual contact. And of those that were closely related, in 76% we were able to tell the direction of transmission. Now, if you look at the population-based sample, and this is what we're really interested in, the figure was dramatically different. Illustrating one of the complications or challenges in working on a population-based sample, where we don't have just selected individuals where we know that something likely happened because they were concordant positive, but we're just looking at the population-based sample. So the most striking feature is that this separation property disappeared, so phylogenetic linkage was not clear-cut. And in fact, based on the female-female pairs that I mentioned earlier, we estimate that up to 35% of the linked pairs in transmission networks may not represent direct transmission networks. However, of those that were close, so in this light blue area, 78% had strong evidence that transmission occurred. So we then continued, our next aim was to validate our phylogenetic inferences into the direction of transmission. And the idea was to cross-validate our results against clinical data and epidemiological data that indicated one direction of transmission. So we looked at zero-discordant pairs. We have, for example, here at the top, so the x-axis shows years relative to the date when both partners were positive. So 0 is when the second partner turned positive. And so the female was found positive 3 years prior to the time that the male was found positive, and the male tested negative while the female was positive already. So this is the crucial information. So meaning that the male couldn't have infected the female. That can only have, if this is a transmission pair, can only have infection could only have spread from the female to the male. Similarly, we had CD4 data, CD4 count data, and green indicates that here that the male had a CD4 measurement above 800 cells, and red indicated that the CD4 measurement was very low, below 400. And in that case, we took this as evidence that, again, infection went, the clinical data suggested infection from the female to the male because the female had already progressed much further than the male. So taking these two independent epidemiological and clinical data sources, we had in total 71 phylogenetically linked pairs with such data. Of those, we found that 46. So then we compared the phylogenetic inferences against our epidemiological assignments, and in 46 pairs, these were consistent. In 16, we couldn't tell the direction of transmission phylogenetically, and in 9 cases, we made the incorrect assignment using phylogenetics, and this corresponded then to a false discovery rate of 16 points. So the conclusions from the first part are then that deep sequence phylogenetic analysis can be conducted at the scale of population-based surveys using phyloscanner. Linkage analysis using deep sequence reads have similar limitations compared to using consensus sequences, in the sense that we still, even using deep sequence data, we were not able to prove transmission between two individuals. And finally, the direction of transmission could be inferred in the large majority of closely related pairs with a relatively small error. And we then hypothesized that this error is of course substantial when we're looking at every pair individually, but for population-based analysis, this error is relatively small. So I'm now coming to the results of our study. In total, as I said, we had 293 source recipient pairs that we reconstructed. I'm focusing now here on, I'm showing the pairs that had a source in inland communities and a recipient in inland communities. And the colors indicate the migration status. So the lighter green says that the source case was resident in inland communities, and the dark green case said that that person had recently moved from an inland community. One of the first and striking observations was that out of the 293 transmission pairs that we could reconstruct, 265 were not between inland and fishing sites. 91% were not crossing fishing and inland communities. And this suggested to us that the transmission networks between inland communities and fishing sites were largely distinct. And when we looked further, in total we found 7 transmission pairs, so this was 2.3% from fishing to inland, but 3 times as many from inland to fishing. And now this analysis at that time sort of contradicted what we hypothesized initially. So we had thought that the fishing sites, so this is what we had hypothesized, that the fishing sites would be major sources of inland infections, and in fact, what that unadjusted data showed to us was that it was actually the opposite. When we tried to substantiate this result further, one of the concerns that came to the fore was that we had substantial differences in participation and sequencing rates across the communities. And here I'm showing this on the x-axis for the communities, and the y-axis shows the number of sequenced individuals amongst those that were found positive in the survey. And the blue bars show the proportion or the sequencing rate amongst the males, and the pink amongst the females. So you can see how this varies across communities and also systematically between males and females. And we were concerned that these differences could lead to bias in our inference. And as consequentially, we tried to find statistical analysis to adjust for these participation and sequencing differences, and we used a tool that appeared in 1997, which is called Bayesian data augmentation. I'm probably not going to go into details on this, but the idea was to adjust our estimates for known sequence and participation bias. And this figure summarizes that after adjusting our analysis, we essentially reached the same conclusions. So our estimates now with the confidence intervals are pretty much the same as before, 3 times as much overall flow from inland communities to fishing sites than vice versa, largely distinct networks, and also some inflow from external. Overall, when we looked at the ratio of transmissions from inland to fishing versus fishing to inland here on the x-axis, we clearly found significantly more transmission from inland communities to fishing sites. So this was significant. However, when we stratified by migration status, so when we looked at resident sources that were resident and sources that were out migrants, we also found significant differences. So the flows from inland to fishing were substantially higher than vice versa amongst out migrants, and this indicated to us or suggests that population mobility at this small scale is really a major factor that turns the fishing sites into epidemiological sinks in the transmission. So implications for geographic targeting. This is just a cautious case study looking at one example of 4 fishing sites in a specific corner of Lake Victoria. So we should be cautious in interpreting these results. But it does show that hotspots are not necessarily the main sources of new infections in neighboring low-risk areas. Of course, populations in hotspots will directly benefit from geographic targeting of combination HIV prevention. However, the indirect benefits that are often stated or with which geographic targeting is justified aren't necessarily the case and may be limited in our particular setting. So going forward, bigger lessons from the study are that mapping may not necessarily be enough for determining hotspots that drive transmissions in neighboring areas. We feel that as a next step, a more comparative assessment of hotspots on their contribution to the broader spread of HIV should be a priority. So we don't, I don't think it's needed to look at 20 or 30 different hotspots, but for example, just within PANGEA consortium, looking at different settings and the hotspots in there, and trying to sort of, in a way, maybe repeat this kind of analysis and try to assess where the hotspots in different settings are really indeed the drivers of transmission to neighboring areas, or if they are not. And maybe this also leads to a characterization of what potentially might make a hotspot a high prevalence or high incidence area. What factors might make that a driver of transmission to neighboring areas. And finally, similar analysis could help allocation of limited resources when they're really limited, by prioritizing hotspots for geographic targeting that have a large impact on the epidemic. And with that, I'd like to come back once more to my acknowledgements. The Rakai Health Science Program, the program staff, the study participants with whom this study would never have been possible, Kate for her ongoing support and substantial contribution over the past 2 years and to this analysis. Also Matthew and Chris, of course, because they're primary drivers behind Phyloscanner. Tanya, who did much of the read mapping that underlies all the analysis, and Christoph for in particular also for lots of valid criticism throughout. Thank you very much.